why is that they're targeting on KRAS? Okay, so basically with the wild type KRAS, okay, KRAS is linked with the marker, the so-called epidermal growth factor receptor. Okay, at the surface level. Okay, this is the EGFR. Okay, this marker, epidermal growth factor, there is the presence at the surface level. But the expression is manipulated by the activation of this KRAS mutation. Okay, so ideally, when they want to kill the tumor cells, they want to stop the tumor cell from growth. So ideally, they will block this EGFR expression. But somehow, remember the concept of resistance just now? Not all patients will benefit onto this targeted treatment, where that treatment, okay, that patients only
Hello.
exist in one. Okay, it exists in more than that. It got multi types, multi variants of the KRAS mutation. So, for example, G12A, G12C, G12D, G12V, and many types. Okay, so you can see that the very frequent one is G12A, 12C, and 12T. Okay, and the frequency of the KRAS mutants, they are different according to the type of cancer. So, for example, you can see that G12V, D, and C, they are prominent to the lung. Okay, but the different frequency is different. Okay, for example, between lung and colorectal, for the lung, the very prominent one is G12C, with the colorectal is G12D. So can you see that how the variety of the mutation frequency develop a different mechanism signaling on the cancer types? Okay, so they got a different cancer types here. Different cancer types, they will have a different mechanism of signaling, which due to the different frequency of the mutations. Okay, so with that, okay, when we did NGS, for example, NGS, so because we cannot apply Sanger sequencing at the moment, because of with the Sanger sequencing, you can only use one particular mutation. Am I right? But with this NGS, okay, or we call it targeted NGS, for example, you can see how the frequency of mutations happen, as well as how they co-detected, co-mutated with some other gene mutation. Okay, for example, in here, this is the heat map. Okay, and that you blast together with the cancer genome atlas. So with this pancreatic adenocarcinoma, they run it among 122 samples. Okay, so basically you can see that the blue font here, the blue font, are the co-mutated with KRAS. So KRAS G12D, KRAS G12R, KRAS G12V, KRAS as well as TP53. So meaning that this particular gene, they are mutated in that pancreatic adenocarcinoma. Okay, and then with the red font here, this is a panel genes here. So you got the red font here, which is contra-mutated. Some patients, they have this mutant, some may not. Okay, and of course they provide the black font here, the black font, the black color here, they represent as a highly mutated, but they are not correlated with the KRAS. Okay, so in here, okay, what this can summarize based on the study here, based on the analysis, that KRAS was mutated in 84, which is 102 samples, patients out of 122 patients, pancreatic adenocarcinoma samples. Okay, 102 in here, up till here, they have this KRAS mutation. Okay, so in here, this is the non-mutant. 20 have no KRAS mutation. Okay, so the most common mutation of KRAS is G12D, G12V, and G12R, which is indicated here. And then they got also two well-known tumor suppressor genes such as TP53 and CDKN2A, CDKN2A, and also TP53, meaning that co-mutated, the mutation happened at the same time. Mutation happened at the same samples. Okay, and then TTN, this gene TTN, it was mutated in 21% here of the sample, but this mutation were not correlated with the mutational status of the KRAS. Okay, so this TTN, for example, so KRAS, okay, TP53, SMAD4, CDK, and 2A, there are type of genes that has been correlated with the KRAS mutation. Okay, it's the same thing happen. So the integration of the mutation, okay, was studied according to the progression of the tumor. So for example, transition from normal to the pre-cancerous type. Okay, so they require to have this KRAS mutation, and then CDKN2A, okay, and then followed by the CDKN2A, and then at the later stage, they may require some other TP53 mutation and also SMAD4 mutation, which continuously progress to become invasion and also metastasis. So meaning that when you do the analysis from NGS, ideally you not only you see the frequency of the KRAS mutation on its own, but you can also see the co-occurring mutation where when a clinician or oncologist need to design the treatment option, they will consider the target on the KRAS, CDKN2A, TP53, as also SMAD4A. And sorry, SMAD4 gene mutation. Okay, so that's about the idea about how they design the treatment option. Okay, so yeah, for example, the treatment. Okay, I think I mentioned about this before. All right, so you got this anti-EGFR. Okay, ideally EGFR exists at the surface level. Okay, and then at the surface level, when you apply this tyrosine kinase inhibitor, block EGFR, so supposedly we understand that this TKI can kill the cells, but perhaps when the mutation happen, okay, it causes increased signaling despite upstream EGFR inhibition. Okay, so the nuclear signaling abnormally increase. So definitely you need to design treatment that targets on these downstream signals. Okay, so this is the intracellular targeting of RAS. Okay, very complex. Okay, so not only targeting at the upstream level, but they go down here to targets on um, the downstream of the KRAS mutations. Okay, the RAF, the MAC, the ERK, PI3K, and AKT. So you can see the type of inhibitors, they already classified them. For example, tri-complex inhibitors, the mutant KRAS inhibitors, so on and so forth. Okay. So ideally, in order to target to stop the tumor, okay, you cannot rely solely on one particular treatment, one particular one particular treatment agent, but you need to consider many type, many multi targets of the treatment. Okay, so yeah, I think we reached into the conclusion here. Okay, the conclusion is that the modern drug development methods require definitive knowledge of the molecular targets and mechanism of action in disease pathology. And of course, it's about to generate a precise knowledge of the complexity of the disease pathogenesis, okay, which we should explore through the integration of the multi-omics methods. Okay, so if you're aware that in my topics, I don't touch more on the analysis, okay, because I'm sure that it 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 is quite complex, okay. But for me, it should be enough to show to you the application wise how from a big data, okay, you can actually get the somehow the idea about that progression of the tumor, the characteristics of the tumor. Okay, so the high throughput omics approaches in analyzing complex biological samples generates massive data. Okay, of course, when it generates massive data, you may need to have not only several, multi type of the pipeline, okay, bioinformatic pipelines that need to help to analyze, to filter the data until you can fully interpret that data. Okay, so the interpretation and curation of the complex data sets are very challenging and require detailed knowledge of the computational and biological system. Okay, and I'm sure that with the computational, which I don't cover in my lecture, you may need to have I think I believe that you may have some other lecture regarding to the bioinformatic tools, right? Okay, so of course, in terms of an integrated omics platform, with the integrated omics platform, they will provide novel insights into cellular system biomarker discovery, as well as it helps into targeted therapeutic, which is the topic of ours lah today. Okay, so here I'll show you here the yeah the diagram. Okay, the flow. How the breast cancer. This is the cohort, the breast cancer cohort. Okay, and then of course the cancer is increasing, and we need to design a treatment. The drug resistant, even the treatment has been designed to stop the cancer cases, it still provide the drug resistant. And of course, in order to cater with this problem, the challenges, okay, we apply this multi-omic approach, okay, because of we need to identify the perfect drug targets for that particular cancer. And this we're considered as a personalized medicine because of why? Because of we generate the design, okay, we design that particular treatment based on profiling data that we have from the patient itself. Okay, so that will conclude our lecture today, right? Is it okay we stop now, or you still need 1 hour to complete 1 hour? Can you guys write down your full name here? Sana Yasin is push button. Okay. Oh. Okay. Who is actually scribble something in this slide? Blah blah. Go say you. Go say you. Sana. Okay. So you guys were asking me what are the requirement for this assignment? What is that? What question is that? I don't understand. Requirement for this assignment? When is the submission deadline? Okay, you know what? You have 1 hour left. Why not you spend your time, go and search for that abbreviation, and then you can submit to me after that.
Doctor, like only the abbreviation and the function of that abbreviation, isn't it?
Yes. Okay.
Doctor, like from 29 to 33, the slides.
29, 23. Yeah. The slide 29 to 23. Submission through eLearn. Oh, how do you normally do that? Through eLearn, right? E-learning. Yeah, you can do that as well through e-learning. We do not have the slides currently with us. Oh, okay. All right. Hang on, yeah. You receive. Could you upload the slide for us to refer to? Of course, Yan, I will do that. Hang on in a minute. I need to. Can I share through e-learning? Yeah, but I need to. How do you normally complete your attendance? Also in e-learning. Or there's no attendance to be taken? Attendance, e-learning. Stop sharing. So this e-learning is what is the topic? G turn terkini dalam Omics, current trends in Omics. It is current trends in Omics. The current trends quiz. Where's my name? They got this artificial intelligence. Yes. Where is my name? Pharmacogenetics. Siti Nora, where are you? Here we go. Omics no Fatmawati predictive 3 hours. Guys, how do you normally dah dismiss ada kelas. Alamak. Yalah, tak apalah. Keluar juga dulu ni. Okay. Okay. Save as. Save as omics approach omics for therapeutics approach for therapeutic approach. Desktop. Save it. Save. Hiya. Where? Astaghfirullahalazim. Save. Mana download ni? Download the Onyx. We call it the Onyx. Then we call it the Onyx. And get rid of the Onyx here. Mamma mia. Yes. All right. Yes. Hello. Assalamualaikum. Yes, I am. Yeah, yes, yes. Okay. What's the problem with the assignment? Okay. So you guys, meaning that you guys cannot do it during this time, right? 11 to 12. Because of you don't have these lecture note. I am trying to figure out how am I going to do it on e-learning. Excuse me for that. But then, okay, it's not necessary to submit during this time lah. In that case, details on the assignment. Yeah, yeah. What are the requirements? Yeah, you are the one who asking me just now, right? The requirement. What do you mean by requirements? Sorry. No, it's okay. What do you normally do with your assignment normally? Mm-hmm. So I already gave you what I want. Uh-huh. Oh no, there's no such thing of doing that. Why you guys are so particular on that? Okay. So it's just assignment. It's just a normal assignment. So just based on the question just now that I gave you just now. So you do some search on it, okay, and then type it and give it back to me. There's no specific. When the deadline is okay, since I thought that I can give you guys do it within these hours, within this hour, but okay, Friday, yeah. How many marks? I have to discuss with Dr. Fatma because Dr.